Today we will discuss the final figure that has been presented in this paper that we are discussing for the past few days. So in this video they connect their experimental studies, experimental results that they got from their previous experiment with the clinical data that are available for the pancreatic cancer cell patients. So far from their previous experiments they come to a point that this CD24, CD44 or SLC16A1 plays a role towards this anchor independent properties of growth and invasion in the pancreatic cancer cells derived patient because of the cell transfer between macrophages and pancreatic cancer cells. So let us start with this figure. So in the panel A they compare the they check the survival rate of the patients that has high CD24 or low CD24 or high CD44 or low CD44 and high SLC16A1 and low SLC16A1. And in this experiment or in this analysis they find out that patients that have high CD24 or low CD24 does not have too much difference in the survival rate. However, the patients that has high CD44 or high SLC16A1 has a lower survival rate compared to the patients that has low CD44 or low SLC16A1. So this result shows that CD44 and SLC16A1 might be important for the prognosis of the pancreatic cancer cells. Thereafter they check the pathways that are dysregulated in this kind of knockout cells in the patients that has this high amount of these proteins. And what they find out is that for the high CD24 they have this high fatty acid metabolism or this xenobiotic metabolism. However, for the CD44 they see the high hypoxia or estrogen response, and the apoptosis rate are also high in the high CD44 patient samples. And for this SLC16A1, high SLC16A1, they see that the pathways that are related to migrations are high, such as mTOR1 signaling and MIC targets and G2M checkpoints. These are high in cells. The GSA analysis also we can see that the higher pathway shows a significantly higher values and when these data are compared with the wild type pancreatic cancer cells, and the top two pathways are shown for each category in this panel that we can see it. For example, CD24 they show it for fatty acid metabolism and cholesterol homeostasis, and for this CD44 they show it for hypoxia and this TGF beta signaling, which is the characteristics for the migrations. And for SLC16A1 they show it for this uh, high high MIC target 1 and hallmark for glycolysis. This is the characteristics for this metabolism pathways, upregulation of metabolic pathways. Thereafter they after validating that it's the CD44 or SLC16A1 might play a role, they again decided to they want to go further and to check whether different combinations of these genes plays a role towards the survival of the patients. So in the panel C they check for CD24 high and CD44 high, and they see that growth rate are similar to the CD24 high and CD44 low. That can be seen here. However, for the CD24 low and CD44 high, they see that lower survival rate, and only the CD44 is high. And also when the CD24 is low, and the CD44 is low, they see the higher survival rates, so indicating that it's the CD44 plays an important role. And for the SLC combinations, the CD24 high and SLC16 high, they see that lower survival rate. However, when the CD24 is low and SLC16A1 is high, they see that the survival rates are also decreases further. But for the CD24 high and SLC16A1, they see that more survival rate, whereas when the SLC16A1 and CD24 is low, the survival rates are further increases. And in the last combinations of CD24 with CD44 with SLC16A1, they see that when the both are high, they see that lower survival rate, and when the CD44 is high and SLC16A1 low, they see that survival rates are further decreases in the yellow region, indicating the CD44 is has a much higher contribution towards pancreatic cancer cells. And the third case where CD44 is low, SLC61A1 high, they see that the survival rates are lower, but the data is not statistically significant. The difference is not statistically significant as the number of samples patients are low that are analyzed in this experiment. And when the both are low, they see that survival rates are pretty high. So all this indicates that the CD44 and SLC16A1 could be the prognostic marker for the pancreatic cancer cells. And if any patient has high amount of this proteins, then there should be a high probability of the patients having pancreatic cancer cells, or they are affected with the pancreatic cancer. This is what this paper has finally made a point in the end, based on their in vitro analysis and compare it with the clinical data. If you need to understand clinical data, if you need to understand more about their experiments in detail and how they have conducted their analysis, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.